Is the development of the urinary system. The competency is about the development of the urinary system. Embryologically and anatomically, the urinary system and the genital system are closely associated in early stages of development. Both develop from a common mesodermal ridge along the posterior abdominal wall. Excreted ducts of both systems initially enter a common cavity called as cloaca. In the male, the urethra conveys both urine and semen. Although separate in the female, the urethra and the vagina both open into a common vestibule. Intermediate plate. Urinary system is derived from intermediate plate or mesoderm lying between the paraxial mesoderm and the lateral plate and the cloaca. Urinary system develops in a craniocaudal direction in successive chronological steps. Definitive kidney or metanephros is preceded by two transitory structures. Pronephros is a rough draft and is rapidly replaced. Mesonephros reaches complete development but later predominantly regresses. Remnants of the system are incorporated into the urogenital system. Nephrogenic cord. It is the intermediate mesoderm that migrates ventrally, loses its connection with the somites. This longitudinal mass of nephrogenic mesoderm on each side of the body becomes the nephrogenic cord. The nephrogenic cord undergoes metameric segmentation into nephrotomes. Nephrogenic cords give rise to renal tubules of the kidney. Pronephros, it differentiates at the end of week 3 in the cervical region and is non-functional. It disappears at the end of week 4. Currently, it is thought that the pronephros disappears completely, leaving no vestiges. It is the mesonephros that forms its own collecting duct. Mesonephros or Wolffian body differentiates in week 4, caudal to rudimentary pronephros, and may function as an interim kidney until the permanent kidney is established. The mesonephros regresses in week 8. Mesonephric nephrotomes pass through the following stages beginning in week 4. Mesonephric vesicle formation. Vesicle grows into an S-shaped mesonephric tubule. The tubule lengthens rapidly. There is an enlargement of the vesicle's internal or medial end to form the glomerular chamber opposite an arterial loop from the aorta. Arrangement of the glomerular chamber around the glomerulus. The chamber forms a double-layered cup, the Bowman's capsule. The capsule plus the glomerulus forms the mesonephric or renal corpuscle. The external end of the tube opens into the mesonephric or Wolffian duct. Mesonephros begins to regress towards the end of the month 2. By the beginning of the fetal period, most of the mesonephros has degenerated and disappeared, except for its duct and few tubules near the gonads, which persist and are annexed as genital ducts in the male and form vestigial remnants in the female. The conodal primonium develops at the internal anterior side of the mesonephros. Metanephros or permanent kidney. Metanephric blastema, a non-segmented homogeneous mesodermal mass, enters into active phase of differentiation at the beginning of month 2 and begins to function about 3 weeks later. The urine formation continues actively throughout the fetal period. Urine combines with amniotic fluid, which the fetus drinks and is absorbed by the intestine. The fetal kidneys regulate amniotic volume. If the kidneys are absent, amniotic fluid volume is abnormally small, leading to oligohydramnios. There is no need of prenatal function of the kidneys because metabolic wastes are transferred via the placenta to the mother. But the fetus would die prenatally without the kidneys. Metanephric differentiation begins when the metanephric blastema is penetrated by ureteric bud, which arises from lower portion of the mesonephric duct. Ureteric bud arises from the lower end of mesonephric duct rapidly reaches and penetrates the metanephric blastema, which in terms forms a cap over the bud. The bud enlarges and forms the beginning of the renal pelvis. It then develops two diverticula, the cranial and caudal, the future major calces. The stalk itself becomes the ureter. Minor calces give rise to collecting ducts and tubules which penetrate farther into the metanephric mass. Collecting tubules in turn further subdivide and in the process compress the blastema eccentrically. They subdivide until about 13 or more generations of tubules are formed. A complete system of branching tubules forming a renal lobe results from each tubule of the first generation. While more and more new tubules arise on the periphery of the blastema, the major calcis absorbs the ducts of the third and fourth generations, which are transformed into minor calcis. The tubules of the fifth and successive generations form the definitive collecting tubules of the adult kidney. Minor calces receive the papillary ducts of the tubular system. The terminal end of the collecting system opens at the pyramid of the apex of the pyramid. Ureteric bud thus gives rise to the entire collecting system of the kidney, namely the ureter, renal pelvis, calcis, papillary ducts and the collecting ducts. Metanephric blastema develops from intermediate mesoderm. It forms a solid mass of tissue. Development of its collecting ducts is formed by the developing ureteric bud. The tubular branching and bud compresses the blastema eccentrically and fragments into tissue caps or cluster of mesenchymal cells. At end of the cap, metanephric spheroid arises, which undergoes the transformation. Namely, there is a spheroid stage, vesicle stage, stage where vesicle elongates in an S-shaped and finally vesicle opens into the collecting tubule. Here the differentiation is elaborate and leads to formation of functional units or nephrons, glomerulus plus the tubular system. It includes Bowman's capsule, the proximal distal convoluted tubules, the loop of Henle, the glomerulus and the Bowman's capsule make up the renal corpuscle. The distal convoluted tubule join the arching collecting tubules and become confluent. Metanephric blastema thus gives rise to entire excretory system. Each uniferous tubule has two parts, the nephron from metanephric mass of mesoderm and collecting system from the ureteric bud. By 3 months, the nephrons already formed are anatomically and histologically identical to those of adult kidney. At this stage of development, the kidney begins to make urine, even though it has not acquired as at its total number of approximately 1 million functional units. The functional units grow in number by concentric layers throughout the prenatal life and are laid down until about term. No nephrons usually develop after birth except in premature infants, but the existing nephrons complete their differentiation during infancy and increase in size until adulthood. Thus, the increase in the kidney size is due to hypertrophy and not to an increase in the number of nephrons after birth. Thank you.